Antimicrobial agents and antimicrobial resistance is the today's topic. At the end of the session, students will understand the classification of antimicrobial agents, intrinsic and acquired antimicrobial resistance. Antimicrobial agents are classified in various ways like antibacterial, antifungal, antiparasites, and antiviral agents. And according to the ability to kill or inhibit the microorganisms, bactericidal and bacteriostatic. According to the source, antibiotics and chemotherapeutic agents, and according to the site of action and usage, disinfectants, antiseptics, and antibiotics. So according to the chemical structure, the mechanism of actions, this diagram shows the first is cell wall, which are acting again on the cell wall, bacitracin, which is a topical antibiotic, which is used for identification of Streptococcus pyogenes, group A streptococci. Then glycopeptide antibiotics, phosphomycin, which is used for urinary tract infection, vancomycin, which is a drug of choice for MRSA, and beta-lactam group of antibiotics. All these act on the cell wall. Another group of antimicrobials is inhibiting the protein synthesis. There are two types, 30S inhibitors, aminoglycosides, tetracyclines, and tigecycline. 50S inhibitors are chloramphenicol, clindamycin, linezolid, macrolides, and streptogramins. Then those antimicrobial agents acting on cytoplasmic membrane, polymyxin B and daptomycin. Polymyxin B and daptomycin. Nucleic acid synthesis inhibitors are fluoroquinolones, which suppress the DNA gyrase enzyme, metronidazole, which suppresses the DNA synthesis, and rifamycins, which will decrease the RNA polymerase enzyme. Then another group of antimicrobials, antibiotics that is inhibiting the metabolic pathway, folate pathway, sulfonamides, then trimethoprim, and combination of both, that is cotrimazole. Now various antimicrobial agents, the examples are given here, and spectrum of activity and mechanism of resistance is given here. So you can see penicillin derivatives, penicillin G, aqueous penicillin G, procaine penicillin, benzathine penicillin, and penicillin V. So which are acting on the gram-positive bacteria, Streptococcus pyogenes, Pneumococcus, Corynebacterium diphtheria, Clostridium tetani, Clostridium perfringens. Others, it also acts on meningococcal infection, gonococcus, and Treponema pallidum. What is the mechanism of action? Drug inactivation by producing beta-lactamase enzyme, alteration of the target site, penicillin binding protein alteration, and decreased permeability. Another is penicillinase resistant penicillins. Example is methicillin, oxacillin, cloxacillin, dicloxacillin. These are the penicillinase resistant penicillins. These are they again inhibit the cell wall synthesis, same as penicillin, plus penicillin is producing Staphylococcus aureus, they are effective. Then another group is aminopenicillins, ampicillin and amoxicillin, same as penicillin, plus the activity is against Enterococcus faecalis, Escherichia coli, Klebsiella, Helicobacter pylori, Salmonella. Resistance also reported against this, and Shigella, bacillary dysentery. The mechanism of drug resistance, drug inactivation by producing beta-lactamase enzyme, alteration of target size, and decreased permeability of the antibiotic. Then anti-pseudomonal penicillin. Anti-pseudomonal penicillin, carbanicillin, ticarcillin, and piperacillin. So again, cell wall inhibiting these are, and same as aminopenicillins, spectrum of activity, same as aminopenicillins, and in addition to that, pseudomonas also. They are acting on the pseudomonas. Then beta-lactam and beta-lactamase inhibitors. The aminopenicillins plus anti-pseudomonal penicillins plus clavulanic acid or sulbactam or tazobactam or avibactam combination. So The spectrum of activity is same as aminopenicillins or anti-pseudomonal penicillins spectrum plus beta-lactamase producing bacteria. Again, the drug of action mechanism of resistance is same. Then come to cephalosporins. Again, they are acting on the cell wall, inhibit the cell wall synthesis. First generation cefazolin, cephalexin is used for the treatment of Staphylococcus aureus, Staphylococcus epidermidis, gram-negative like E. coli and Klebsiella. Then second generation cefoxitin, cefaclor, and cefuroxime. So the spectrum of activity is same as the first generation plus gram-negative activity is increased and increased anaerobic activity. Cefoxitin and cefotitan, they are having increased anaerobic activity. Then third generation cephalosporin, ceftriaxone, cefotaxime, ceftazidime. Decrease activity against gram positives compared to the first and second generation and increase gram negative activity. Some are active against the pseudomonas. For example, ceftazidime. We are testing the antibiotic susceptibility against pseudomonas for ceftazidime. Then fourth generation cefepime, cefpirone. There is a good activity against the gram positive and gram negative bacteria, including pseudomonas. Mechanism of resistance is extended spectrum beta lactamases. Now come fifth generation ceftobiprole and ceftaroline. These are the same as fourth generation cephalosporins and MRSA. Only beta lactam effective against methicillin resistant Staphylococcus aureus. So fifth generation cephalosporins is the only beta lactam which is effective against methicillin resistant Staphylococcus aureus. So only fifth generation cephalosporins are effective. Then carbapenem, imipenem, meropenem, doripenem, ertapenem. So broadest range of activity against gram. Most bacteria include gram-positive cocci, Enterobacteriaceae, Pseudomonas, Listeria, anaerobes like Bacteria specialis, and Clostridium difficile, MRSA also, and Mycoplasma. The mechanism of resistance, they are also not spared from the drug resistance, produce carbapenemesis and efflux pump. Then monobactam, that is the astronam, which is effective against the gram-negative rods. Mechanism of action is production of ESBL. Then beta-lactam plus beta-lactam lactamase inhibitors. Ampicillin sulbactam, amoxicillin clavulanate, cefporazone sulbactam, ceftazidime, avibactam, ceftorozone, tazobactam, piperacillin tazobactam, meropenem, abrobactam. Same as spectrum of respective beta-lactam drug plus active against beta-lactamase producing bacteria, and they have excellent anaerobic coverage. So it's excellent anaerobic coverage. So therefore, suppose if you have started piperacillin tazobactam, so there is no need of starting metronidazole simultaneously for anaerobic coverage. Otherwise, that is considered as redundant use or unnecessary use of overlapping spectrum of antibiotics. Then come glycopeptides, vancomycin and teicoplanin, active against most gram-positive bacteria, including MRSA and Clostridium difficile infection. And the mechanism of action is alteration of the target. Then come fosfomycin, inactivate the enzyme UDP, N-acetylglucosamine 3 enol pyruvyl transferase, also known as MUR, required for cell wall synthesis. Active against urinary tract pathogens, against both gram-positive, gram-negative bacteria such as Enterococcus faecalis, E. coli, etc. So it is helpful for treating the urinary tract infection. Mechanism of action, alteration of the target producing enzyme that inactivate fosfomycin. Then comes bacitracin, topical gram-positive coccal infection. Bacitracin is used for as a differentiation disc for identification of Streptococcus pyogenes. The role is not defined, fortunately. Then protein synthesis inhibitors, 30S ribosomal subunits, aminoglycosides, bactericidal irreversible binding to 30S, gentamicin, neomycin, amikacin, tobramycin, streptomycin. Gentamicin, neomycin, amikacin, tobramycin, and streptomycin. Anaerobic gram-negative bacteria often used for empirical therapy in adjunct with third-generation cephalosporins in respiratory infections, meningitis, and subacute bacterial endocarditis. Mechanism of resistance is drug inactivation by aminoglycoside modifying enzymes, decreased permeability to gram-negative outer membrane, and decreased influx of the drug. Then tetracyclines, it is a bacteriostatic, bind to 30 S subunit of ribosome and block tRNA attachment. Tetracycline, doxycycline, minocycline, demeclocycline. Helpful for treatment of rickettsial infection, chlamydia, mycoplasma, spirochetes, Yersinia pestis, Brucella also, please remember, Haemophilus ducrei, Campylobacter vibrio cholerae. How is the mechanism of resistance? Decrease intracellular drug accumulation, active efflux, throw it out, ribosomal target site alteration. Then glycycyclines, tigecycline is the example, active against Staphylococcus, Enterococcus, Acinetobacter, and E. coli. The mechanism of drug resistance is active drug efflux pump. Chloramphenicol, helpful for Haemophilus influenzae, pyogenic meningitis, brain abscess, anaerobic infection, enteric fever. Previously, it was favorite drug, but now not used due to drug resistance. How is the mechanism of action? Drug inactivation by producing chloramphenicol acetyl transferase enzyme, altered membrane transport. Then macrolides, erythromycin, azithromycin, clarithromycin. Macrolides are azithromycin, erythromycin, and clarithromycin. Spectrum of activity: Streptococcus, Haemophilus influenzae, Mycoplasma pneumoniae, alteration of the ribosomal target, and active efflux of antibiotics. Then comes lincosamide, clindamycin, and lincomycin are example. Bacteroides praesalis, Staphylococcus aureus, toxic shock syndrome, and excellent bone penetration is very important. So orthopedic infection. The mechanism of action is alteration of the target site, ribosomal methylation. Then oxazolidones, linezolid, oxazolidones. Resistant gram positives like MRSA, alteration of the target site. Streptogramins, quinpristin, dalfopristin, Streptococcus pyogenes, Staphylococcus aureus skin infections, MRSA infections, VRE infections. Alteration of target, active efflux, quinpristin, drug inactivation, quinpristin and dalfopristin. Then nucleic acid synthesis inhibitors, fluoroquinolones. They inhibit the DNA gyrase enzyme A subunit and topoisomerase, thus inhibiting DNA synthesis. Nalidixic acid, coliform gram-negative bacilli, fluoroquinolones first generation, norfloxacin, ciprofloxacin, ofloxacin. And second generation is levofloxacin, lomefloxacin, moxifloxacin, and sporfloxacin. So for treating the Enterobacteriaceae, E. coli, Klebsiella, Enterobacter, Salmonella, Shigella, Proteus, Yersinia. Others we can use for Neisseria, Haemophilus, Campylobacter, Vibrio cholerae, Pseudomonas, Staphylococcus aureus. How is the mechanism of action? Alteration of the drug target, mutation of DNA guide genes, poor transport across the cell membrane. Then the DNA synthesis inhibitors, nitroimidazoles, metronidazole, trinidazole, and secnidazole, which are used for anaerobic organisms. So decreased drug uptake, active flux, decreased drug activation. Then nitrofuran. Nitrofurantine is the example used for urinary tract infection, E. coli, Klebsiella, Enterococcus. Altered drug activating enzyme. Nitrofurantine is very good oral drug, but we should identify that it should not be recommended for, it should not be tested for uh, uh, Proteus vulgaris. It should not be used for Proteus vulgaris, nitrofurantine. Rifamycin, rifampicin, rifaximin. Mycobacterium tuberculosis, Mycobacterium leprae, non-tuberculous Mycobacteria, Staphylococcus aureus, prophylaxis of H. influenzae meningitis, prophylaxis of meningococcal meningitis. And how is the target mutation in RPOG gene? So we are detecting in gene expert, that is CBNet test. We are detecting the mutation in RPOV gene. Then mycolic acid synthesis inhibitors, INH, used for tuberculosis and latent tuberculosis. So here, how is the mechanism of action? Mutation in enzyme processing isoniazid into active metabolites, CATG enzyme. Then folic acid synthesis inhibitors. It is a bacteriostatic competitively inhibit the enzymes involved in two steps of folic acid synthesis: para-amino benzoic acid folate synthesis is synthesis para-amino benzoic acid under the folate synthesis enzyme. It is formed dihydrofolic acid, and after the influence of dihydrofolate reductase enzyme, it will get converted into tetrahydrochloric enzyme. So folate synthesis is inhibited by sulfonamide, folate synthesis, and folate synthetase, and dihydrofolate reductase, it is affected by trimethoprim. So both they act synergistically, leading to inhibition of folic acid synthesis. So sulfadiazine, sulfacetamide, cotrimazole. So used topically in burn on surface. Cotrimazole is indicated in urinary tract and respiratory tract infections, active against Serratia, Klebsiella, and Enterobacter. So how is the mechanism of action? Production of insensitive targets, dihydropurate synthesis, and dihydrofolate reductase that bypass metabolic block. Then gramicidin, which is useful for topical use against the gram-positive cocci. Mechanism of resistance is not defined. Lipopeptides, daptomycin, bactericidal against gram-positive bacteria, including VRE and MRSA. The mechanism of resistance is not defined. Then polymyxins, polymyxin B, colistin, or polymyxin E, helpful for gram-negative infections. So here the mechanism of action is alteration of lipopolysaccharides, efflux pumps mediate. What is the antimicrobial resistance? So what are the types? The antimicrobial resistance is development of resistance to antimicrobial drug by microorganisms. It can be of two types: intrinsic resistance, acquired resistance. Intrinsic resistance, acquired resistance, emergence of resistance is bacteria that are ordinarily susceptible by acquiring the genes coding for the resistance. Overuse and misuse of the antimicrobial agents is the single most important cause of development of acquired resistance. So use of particular antibiotics gives selective pressure in the population. So the resistance bacterial population flourish. So what are the factors which are favoring? Poor infection control practices, inadequate sanitary conditions, inappropriate food handling, irrational use of antibiotics by doctors, uncontrolled sale of antibiotics over the counters. So how is the selection pressure? Susceptible organism will be killed. So only resistance will be there. So final population resistant organisms. And these resistance organisms will flourish and will cause the infection. Intrinsic has nothing to do with us, but the knowledge about the intrinsic resistance is very important because for releasing the report and testing, we should be careful. So what is intrinsic resistance? It refers to the innate ability of the bacterium to resist a class of antimicrobial agents due to inherent structural and functional characteristics. So Enterobacteriaceae, the members of Enterobacteriaceae are intrinsically resistant to antimicrobials specific for gram-positive organisms such as clindamycin, daptomycin, picoidic acid, glycopeptides, lipo, lipoglycopeptides, linezolid, tedizolid, quinpristine, dalcopristine, rifampicin, macrolides, erythromycin, clarithromycin, azithromycin. But there is exception here. Salmonella and Shigella species are susceptible to azithromycin. You know, Salmonella, we are treating antic fever with azithromycin. Then Klebsiella pneumoniae, same as the Enterobacteriaceae plus ampicillin and ticarcillin. Klebsiella pneumoniae. Citrobacter species same as Enterobacter plus ampicillin, first and second generation cephalosporins, and cefamycins, amoxicillin clavulanic acid, and ampicillin sulbactam. Enterobacter species same as Enterobacter plus ampicillin, first generation cephalosporins, and cefamycins, amoxicillin clavulanic acid, ampicillin sulbactam. Non-fermentative gram-negative bacilli. Non-fermentative gram-negative bacilli intrinsically resistant to penicillins, cephalosporins, that is cephalothin, cephalosporin second generation, cefuroxime, then cefamycins, cefoxitin, cefotitan, clindamycin, daptomycin, fusidic acid, glycopeptides, linezolid, macrolides, quinpristin, dalfapristin, and rifampicin. Pseudomonas aeruginosa, same as NFGNB plus ampicillin, ceftriaxone, amoxiclav, ampicillin sulbactam, ertapenem, tetracycline, stachicycline, cotramoxol, and chloramphenicol. Acinetobacter, same as NFGNB plus ampicillin, amoxicillin, amoxiclav, ertapenem, acetonam, chloramphenicol, and fosfomycin. Stenotrophomonas multiplex, same as NFGNB plus ampicillin, amoxicillin, cefoxitasm, ceftriaxone, cepipam, amoxiclav, acetonam, imipenem, meropenem, ertapenem, polymyxin B, aminoglycosides, chloramphenicol, and fosfomycin. So, a lot of drugs is susceptible. It is having intrinsic resistance. Then gram-positive bacteria intrinsically resistant to astronome, polymyxin BD, colistin, and nalidixic acid. For example, Staphylococcus aureus, same. That is, it is resistant to astronome, polymyxin B, colistin, and nalidixic acid. Enterococcus species same as gram-positive bacteria plus cephalosporins, aminoglycoside, clindamycin, and cotramoxol. So, mutational and transferable drug resistance. Mutational drug resistance, resistant to one drug at a time, multiple drug transferable. Low degree of resistance in mutational, high degree of resistance. Mutational drug resistance can be overcome by combination of drugs, but transferable cannot be overcome. Mutational drug resistance example, combination of drug tuberculosis treatment. Virulence of resistance mutants may be lowered, but here vigilance is not decreased. Resistance is not transferable. Mutational only the vertical transmission from the first generation to next generation, but here transferable drug resistance transferable to other organs. So horizontal transfer will occur by conjugation or by transduction, transformation. But in mutational, there will not be horizontal transfer. So what is the mechanism of antimicrobial resistance? Decreased permeability, preventing the antimicrobials from entering into the cell in seed in Pseudomonas, Enterobacter, Klebsiella species against drugs such as imipenem, aminoglycosides, and quinoline. Efflux pump, throw it out. The drugs are thrown out by the organism. Immediate expulsion of drugs from the cell by thereby preventing the intracellular accumulation of drugs. E. coli and other Enterobacteriaceae against tetracyclines, chloramphenicol. So E. coli will throw out the tetracyclines and chloramphenicol. Staphylococci throw out macrolides and streptogramins, and Staphylococcus aureus and Streptococcus pneumoniae throw out fluorochlorins, flux. Enzymatic inactivation, beta-lactam, all of you know, breaking down the beta-lactam ring. Aminoglycoside modifying enzyme destroy the structure of aminoglycosides. Then chloramphenicol acetyl transferase destroys structure of chloramphenicol. Then come to the modifying the target site, MRSA. It is well known, penicillin binding proteins are modified. Then streptomycin resistance in Mycobacterium tuberculosis due to modification of ribosomal proteins or 16S RNA. Rifampicin resistance in Mycobacterium tuberculosis due to mutations in RNA polymerase. Beta-lactamase enzymes. So capable of hydrolyzing beta-lactam rings of beta-lactam antibiotics, thereby deactivating their antimicrobial properties, produced by gram-positive and gram-negative organisms, plasmid coated, transferred from one bacterium by other, mostly by conjugation, but except in Staphylococcus aureus, where transduction is the mechanism of transfer. So there are extended spectrum beta-lactam is resistant to all penicillins, plus first, second, third generation cephalosporins and monobactams. Sensitive to carbapenems and cefamycins. So resistance can be overcome by use of beta-lactam along with beta-lactamase inhibitor. AMC-beta-lactamase is in addition to the antibiotics for which ESBL producers are resistant. AMC-beta producers resistant to the are resistant to cefamycins, but sensitive to carbapenems. Resistance cannot be overcome by beta-lactam plus beta-lactamase inhibitor combination. Then carbapenem is resistant to all those antibiotics which are AMC beta-lactamase producers are resistant. In addition, they are also resistant to carbapenems. Resistance cannot be overcome by beta-lactamase inhibitors. Important carbapenem enzymes are Klebsiella pneumoniae carbapenems KPC and NDM, New Delhi